Assalamu alaikum students. Welcome back to the online classes of Pharmaceutical Quality Management Theory plus Lab. So today we will discuss about the remaining topics of moisture content determination, and we will also discuss the new chapter that is biological assays. So basically in this discussion we will discuss about method 3, gravimetric method, method 4, dew point, and method 5 of moisture content determination, that is the electrolytic hygrometric method. And also we will discuss about different types of the bioassays that are being performed. So the next method, that is the gravymetric method. Basically, this method is used for determination of moisture content in a solid. So that's why I said this method is used for solid samples. And in basically this method is also known as loss on drawing or LOD. So basically it is based upon change in weight and employed for volatile material. So if you want to check any difference in the quantity between the volatile material present and the non-volatile material, then this method is employed. So in this method heat is applied to remove water from solid sample, and whatever weight difference is there, and that's calculated for determination of moisture content. So the procedure for gravimetric method or loss on drawing method, you have to place and accurately weigh the 10 grams of sample or specimen in a tiered evaporating dish. You have to dry it at 105 degrees centigrade for 5 hours and weigh, means that you have to reweigh after 5 hours, and again after weighing you have to place it again in your oven or heating instrument you are going to use. So you have to continue this drawing and weighing at 1 hour interval until the difference between two successive weights corresponds to not more than 0.25%. So basically after 5 hours you have to continue this drying, and plus after 1 hour weighing it again until the two successive, means one after other weights, the difference is not more than 0.25%. Basically this is the journal procedure according to USP, and the temperature can vary with the sample if it is specified in the individual monograph. So basically it's with a very accurate calculations that it shows can know actually the difference between two last two weights is not more than 0.25%. So basically over there journally that is performed is that you have to continue drawing and weighing process till you are going to achieve a constant weight, means until there is no change in two weights occur, or that's very minute that cannot be calculated. So in that case you are going to perform this procedure, and you have to continue these steps till you are going to achieve a constant weight or a very minute difference between two weights that is not more than 0.25% according to USP. So the next method we have is the dew point method. So in this method, the temperature below which dew is formed is used for analysis of the moisture. So that's why it's called dew point method. So basically in this method, the determination of moisture content is performed using gas specimen, and especially to check the environmental moisture content, this dew point method or instrument is being applied. So this method says it gives measurement of relatively low concentration of water in vapor state in a gas specimen. So it's a very precise when it will going to give you a very low concentration calculation of water that is specially available in a sample of gas, or you can say in the environmental level. So the apparatus that is used for this method is consist of a fog chamber in which the specimen is compressed and observed. So in that fog chamber, you are going to observe your sample till the fog is produced in that chamber. So whenever the sample is compressed and till the fog is produced, you are going to observe it. In successive trials, the pressure is increased and released until minimum pressure that produces fog upon sudden release is determined. So you have to continuously apply the pressure and you have to release that pressure to produce a point where the fog or dew is produced. So that pressure that causes produces fog upon sudden release, that is termed as dew point, and you have to uh, check that point in this method on the instrument. So basically whatever water content is there, that is calculated by the reference to the calibration chart. So by this method, once the fog is produced, the dew point is reached, you can calculate the water content, and by comparing, by checking, confirming it through the calibration chart, you can provide the results. The final method for moisture content determination according to USP, that is electrolytic hygrometric method. So say the process of electrolysis is applied in this method and current is measured. So in this process, basically the electrolysis applied to the water content, and the amount of current that was required, that is applied, that caused the electrolysis of the water is measured, and that provide the reference of the water content available in the sample when you are going to use this method. So it says determination of moisture content in gas and environment, again like dew point method. This electrolytic hygrometric method also employed for the determination of moisture content either in a gas specimen or whenever you are going to check basically the environmental moisture content level, then this method is used. So basically the apparatus uses a sensor cell for water absorption and electrolysis, and a micrometer for measurement of electric current required for it. So a sensor cell and the micrometer, two important parts of this electrolytic hygrometer. So basically the sensor is going to check the absorption of the water and the causing the electrolysis, and a micrometer for checking the current required for the electrolysis of water. So by this way you can determine again water content in a gas specimen, or you can in simple words you can determine the environmental moisture content level by using electrolytic hygrometer. So the next chapter topic we are going to discuss is the biological assays. So it says they are also known as bioassays or biological standardizations or biological evaluation by the different books and the website resources. All terms are going to refer the same thing. So these are the basically assays that are carried out on a living organism and used to determine biological activity of a drug, that is the basically therapeutic and the toxic effect. The biological activity include either therapeutic or toxic or both of them. So basically when you going to use living organism for performing the assay for checking the biological activity, it is termed as biological assays or bioassays. So Further proper definition, it says determination of relative strength of a substance, basically as a drug, by comparing its effect on test organism with that of a standard preparation. Basically, the bioassays are the comparative studies in which you are going to compare the strength, the effect of a drug in test organism, in a living organism, using a proper standard preparation of it. So you are comparing a test or sample material effect with that of a standard officially available standard preparation, to making sure that your drug is sample drug is working properly or not. So test organism classifications, basically according to living organism, the different test organisms that are employed for bioassays or biological assays. They include in vivo biological evaluation, usually in which the intact animals, living animals are used. In vivo bioassays or biological evaluations. Next is the ex vivo biological evaluation or bioassays. Usually in this, the isolated organs or tissues are used, means no need of intact animal. If you want to perform the evaluation in heart, you can take heart of your mice or whatever the specific pigeon specified animal is there. You want to check effect on the heart of any animal, you can only take that isolated organ or tissue and perform that. No need to take whole organism for it. And next is your in vitro biological evaluation. In that, basically cells or the microorganisms are basically used using test tubes and the petri dishes for evaluation. So that's why it says in vitro biological evaluation. So this was the different types of the test organism that are employed for bioassays. Depending upon the availability of the instruments and the material available to you, you can use any type of the organism. Either you can use intact animals, you can use isolated organ, or the tissues if you have the stable um, preparations that make them stable for longer duration, or you can maintain the um, efficacy or their efficiency of your test organism. In the you can use the cell cultures or the micro organism by using in vitro. So main requirement for these test organism is that you must use same strain. If the strain is available, like in case of microorganism, and in case the strain is not available, you have to use the same species and under same gender, in both in vivo, in ex vivo, or even in the in vitro. When you are using intact animals or isolated tissues, along with the same species, you have to choose the same gender, so that there wouldn't be any much difference in the effect, as the biology is not able to remain same and cannot provide you the proper accurate result. That's why you have to choose the same strain in case of microorganism, in case of the cell cultures where the strains are available. Otherwise you have to use same species and same genders. So basically, the objectives of bioassays or biological assays is to check pharmacological effect of new undefined drugs under investigation, to check strength or concentration of the substance, and to check the effect of drugs to the endogenous mediator. So basically, if you are your chemical you have identified, you have discovered, or you have synthesized, you want to check its pharmacological effect, you want to use it as a drug, you have to check, confirm its pharmacological effect. You have to confirm its strength and the concentration by using living organism, and you have to check the effect upon the whatever endogenous mediator hormones or things are there, which means that your drug is going to produce how much effect upon those endogenous mediators. So all these things are the objective of your materials testing uh, things. So you have to check all these points individually as well as collectively. You can use these points for if you are going to use a bioassay. So disadvantages, so there are a lot of disadvantages over there when you are using bioassays or biological assays. Why? Because it's a less precise or accurate method as compared to the physical or the chemical or instrumental methods. So you have to be very careful. That's why you have to use same strain, same species, same gender to make this inaccuracy or less preciseness bit less as compared to the when you are using the many species or many gender many strands in the same test. So you have to use the same thing so to make it little bit more accurate. Whatever the analysis or result is there, it's more time consuming. Obviously biology, you have to wait, you have to um, make a space for the drug to produce an effect upon the living organism. That's why it's somewhat more time consuming, it's more expensive, and sometimes it's need more expertise in the evaluation, in the isolation of the tissues, or even in the applying injections, or in the specific administration technique when you are using animals. So it's more expensive, it's less quantitatively accurate than chemical assay. Obviously assays ke biology nothing remains same. Still if you are using same strain, same uh, gender, still there will be much differences in the results, which may not be because of the chemical nature. It's the biological nature of the organism that produces that is not coming up with that effect that was intended. So as it says, effect on test organism can differ from patient. So basically biological assays are the initial step to check any drug's biological activity. So before moving into the patient or the human participants, you have to check its effect on any living organism. But the problem is that there are chances that as the species differences there, as the too much biological differences are there in humans and in the animals. So it's possible that whatever the effect on test organism are there, that may not be same when that same drug is being tested on the people or patients or the humans during the clinical trial phase. So again you have to be very careful. uh, Technique and interpretation varies with the operator. So obviously when the technique uh, and plus the interpretation of the results, that will be varies with the operator. Different operators use different technique of using same route of administration, and different operators can interpret different results while using the same things. So these are the disadvantages of this method. Okay, if you are using the biological assay, you have to be careful about these things. Okay, these things can happen. So need of bioassay, obviously there are huge disadvantages are there, but still we need to perform the bioassays in the situations uh, where you are dealing with the new drugs. You are going to present your new chemical as a new drug, as a to treat or to for treatment, for prevention, or for diagnosis of any disease or anything. So for that purpose you have to perform bioassay before moving directly into the patients. And second, so it says when the chemical identity of active principle has not been elucidated fully. Means when you are using multiple techniques but it's still whatever the active is there that cannot be properly understood by the chemical nature. So in that case, again, you can perform bioassay to make sure you can make a correlation that with the therapeutic effect and with the chemical nature of the drug. So basically, it says pharmacological action can give you the idea about the chemical nature of the drug. So, and when there's no adequate chemical or physical assays available, means like for insulin, there's no any adequate assays. Assays are available, but not adequate assays are available. So, you have to confirm it through the biological assay. Next is when chemical assay is not valid indication of biological activities. The lack of differentiation between active and the inactive isotopes. So basically, when chemical have different isotopes, some are active, some are inactive. Chemically, they can be identified, but biologically active and the inactive isotope only can be differentiated by using bioassay. So in that case, again, you have to perform bioassay for checking the efficacy or you can say quality of your product. So next is when drug is composed of complex mixture and activity. So like digitally, there have been many drugs are the single constituent, but they have multiple pharmacological effects. So when your drug is very complex mixed, either it's a composed of complex mixture or it's having a complex activity, one drug can produce multiple effects. That again you have to verify the effect or activity by using bioassay. This is the another reason we are using bioassays. um, Next, it says when separation of active principle from interfering substances is impossible or impractical, such as vitamin D from various supplement sources. So sometimes it's difficult to separate the actual pharmacologically active principle or portion from the interfering substances, and you have to use it as a mixture with the along with the other sources. So to confirm their biological activity, you have to perform the bioassay. And next it says when specificity, sensitivity, or practicality dictates the use of bioassay rather than chemical assay, means that your drug demands anyhow you have to perform the bioassay. Then in those all situations you have to perform. So types of bioassays generally, there are two types in journal: the quantitative bioassay and the qualitative bioassay. So in the qualitative bioassays, biological activities, the important indicator, any pharmacological effect that is being shown is measured. It's if it is says it's temperature is there or not. Qualitatively, you can determine. In quantitative bioassay, the quantity, concentration, potency of a drug along with the pharmacological effect is to be measured. So in the difference is only biological indicate activities is being recorded, okay, whether the effect was there or not. And the second is the quantitative, okay, how much quantity was used and how much effect was observed in that quantity. So that is the quantitative bioassay. So further, different types of the main methods. General methods were those. Then there are further main methods available. The one is the graded response, in which it says in these assays, as the dose increases, there is an equivalent rise in the response. The potency of sample is estimated by comparing with the standard preparations. Response curve is like effect of adrenaline on the blood pressure. So these are the types of assays in which the quantity of the drug and the effect level are checked simultaneously. So there was increased effect or decreased effect, or how much adrenaline produced, how much effect upon the blood pressure. So those type of the assays are termed as graded responses, in which the different doses are there, and initially the low doses and then the more doses are used and then compared. Okay, in how much dose how much response was observed, so that's why they are termed as the graded response bioassays. Second is your time dependent. So basically, in this type of the bioassays that are based on duration of the therapy, like vitamins, antibiotics as a curative therapy, is like, yeah, simply you can say antibiotic minimum 3 days and maximum 15 days uh, is like mostly the duration that is recommended for treatment of any particular condition, so that you have to continue the treatment till that duration. If even if still the symptoms are subsided during first day, still you have to continue that intake till 3 days. So in the vitamins or in the antibiotics, that duration is important to achieve the maximum benefits. So that's why these type of the bioassays are termed as time dependent, basically, in which the effect and the amount plus that are usually depend upon the duration of the therapy. That's why they are termed as time dependent bioassays. Next is the endpoint quantile all or none response. So all are the basically the synonyms that are used for the same method. Either you can say endpoint bioassay, either you can say quantile bioassay, or you can say all or none response. So basically, in this method, the threshold dose of the sample is required to elicit a complete or a specific pharmacological effect, which is determined and compared with the standard response. So the example, we have the digitally induced cardiac arrest in pigeons as like lethal dose, and as an effective dose, usually the vomiting is analyzed. So basically, the specific dose is being used, and to check whether that specific effect is produced or not. End point one effect, either response is there or not. Like at the end, the name suggests all or none response, either yes or no, either there is a cardiac arrest by the digitalis or not as a lethal dose, or there is a vomiting is there when you are analyzing effective dose or not. So there won't be like a heart was not proper, the stomach was not proper, still vomiting was not there. Like, it's not like graded response, it's just you are going to observe a single response, whether that response is achieved or not. So that's why it's termed as endpoint quantile all or non response bioassays. So the further description says if the pigeon dies of that, it means that there was cardiac arrest. If there's nobody dies over there, means no any response, means all or none response, like that. Similarly, insulin, if you are analyzing, so insulin induced hypoglycemic convulsion, that is also a type of endpoint quantile all or none response. So if the only hypoglycemia occur but not the convulsion, so it won't be all or none. It will be how much hypoglycemia occur with the insulin, it will be a graded response. But if the hypoglycemic convulsion is achieved or not, only that you are checking, not the glucose level, then that procedure is your endpoint bioassay. So, bioassay methods according to grade point procedures are analysis, include matching or bracketing method, interpolation method, multiple point bioassay. So, matching or bracketing method first of all. So, in this method, an standard dose is bracketed by varying doses of sample till exact matching of sample dose response and standard dose response achieved. So we are making various doses till in the graph the exactly sample dose response and the standard dose response achieved with exact matching, then this method is termed as matching or bracketing. So we are bracketing, dividing into different varying doses, and we are checking the matching dose of the sample that has the same response as that of standard dose response among various doses of the standard 1, standard 2, standard 3, standard. So which dose is having the exactly matching with the sample dose response, we will check that in this method. Next method is your interpolation method. So in this method means interpolate means to estimate, to have an estimated approach to derive any conclusion from the method. So basically in this method it is conducted by determining the amount of sample preparation of unknown potency required to produce a definite effects on suitable test animals or organs or tissues or cells. So these responses are then compared with the standards, and the results are usually expressed in percentages. So plus minus 20% is good, while plus minus 10 usually in the bioassays is thought as excellent. So basically here we are having the estimation, and that is based upon the percentages, okay, how much of the animals have shown a response when the sample or standard doses are given, and then they are being compared. So next method we have is the multiple point bioassays, in which may involve the both interpolation and the bracketing methods. So multiple point bioassays are again are of two types. One is 2 plus 1 or 3 point assay. Second one is 2 plus 2 or 4 point assay. So in 2 plus 1 or 3 point assay, two standard doses are used to evaluate two standard responses, while a single test dose to evaluate a single test response. So total becomes 3. So we are checking 3 doses, 3 responses, among which 2 are of different doses of standard, whereas one is of your test sample dose and or the test sample response we are checking, the unknown we are going to check. So basically in this, two standard doses are given in a ratio of 2 to 3, and an average dose of both standard is given as test. So basically the two doses are given into 2 into 3 ratio. If two parts are given in standard 1 or S1, then three parts are of the doses are available in the um second standard dose, whereas the middle or in between, or we can say the average of both doses are taken as a test dose. That single test dose that is taken, that is tested and checked, is actually in between or average of the both standard doses that are being used for comparison. So simply you can say if 2 mg and 3 mg of standard are given, then approximately your test sample will be of about 2.5 mg. So you have to take 2 mg response, 3 mg response of the standard, and 5 mg response of sample in 3-point assay. Similarly, 4-point assay are 2 plus 2, two standard doses to evaluate two standard responses, while two test sample doses to evaluate two sample responses. So they move went with the parallel comparison because two standards and the two test samples are being compared over there. So basically this chart is the example of 3-point or 2 plus 1 assay or bioassay. So basically you can see in this chart that there are days, day 1, day 2, day 3, and there are groups that are A, B, and C, and the doses given are the S1, S2, and T1, means two standard and one test are sample dose. So basically in this way it says to confirm the crossover studies performed, same procedure is repeated 3 to 4 times in same group but differently. Means each group receive all three doses but basically in different days. So a complete crossover test is there, like in day 1 A group will receive S standard 1, B will receive standard 2 dose, C group will receive test dose, that is the day 1. Okay, similarly on day 2 A will receive the test dose and its response will be tested, B will receive the standard 1 and C will receive standard 2. On day 3 A group will receive standard 2 dose, whereas B will receive the test sample dose and C will receive um, standard first standard dose. So by this way you can confirm okay, the variation in the responses is basically due to the changes in the sample, because all the doses are being tested in all groups that are being used and the compared. So all will receive all three doses in 2 plus 1 or 3-point essays, whereas if it is 2 plus 2 or 4-point essay, then all the groups will receive 4 doses of two standard and the two test sample to confirm their efficacy, potency, dose, whatever the desired outcome or response, or you can say the parameter you want to check. So there are also some other methods that are categorized as other methods for bioassays that include immunological assays, microbioassays, radioimmunoassay, and biotechnology. So these are the other method that utilizes biology or living organism, but they are being mentioned in the specific categories because they use some of specific methods or the tools or materials for their measurements, so they are being classified as the other methods. So immunological assay, it's also known as ELISA or EIA. So either it is ELISA like enzyme linked immunosorbent assay or enzyme immunoassay. So these are the terms and the abbreviations used for the immunological assays. So it's a popular format of wet lab. So it is the wet lab is discussed as it is the type of analytical biochemistry that uses a solid phase enzyme immunoassay for determination of concentration of any substance, usually an antigen in a wet sample or liquid sample. So usually the sample that is available in the liquid form, and that is used in this type of the immunoassay. And basically enzyme based assays are there, so that's why um, we can say the antigen antibody reaction type material is going to determine the concentration of any substance. Either you can use antigen to determine antibody concentration, or you can use any antibody to determine any antigen concentration, or any hormonal level uh, concentration in any patient. So that's your wet lab technology, or you can say the immunoassay based on the enzymes basically. So it's this method, so basically used in medicine and the plant pathology as a diagnostic tool. So obviously in the medicine, in the our field, basically it is used to diagnose the concentration of various things. Either it can be determining the concentration of any antibodies, or any type of antigen, or any hormone concentration in the body, or any allergen concentration. So depending upon the use, either these kits may use the antigens or antibodies, or any reacting hormonal chemicals, or such neutralizing chemicals that can help in the diagnosis of various diseases like HIV, hepatitis, or determination of food allergens in the food. Like in the pregnancy test, usually that the kit is having the anti-human gonadotropin hormone for performing the test. So these are the example in the normal routine life we are using certain kits that uses the immunoassay technique for diagnosis of various diseases or various conditions. So these are the few um, example of it. So basically the kits, as I have discussed, kits are prepared by impregnating either antibodies or antigens. So depending upon the need, either antibodies are available or antigens are available in those specifically designed kits that uses this technology. So next method is microbioassay. So as the name suggests, the bioassay performed by using microorganism as a test organisms. Okay, specifically those tests that are performed on the microorganism. Obviously, they are utilizing the biology or living organism. So that's why they are coming under the biological assays. But as only microorganisms are being employed, so we say the microbioassay. So for this method, it can be utilized for checking potency of antibiotics and vitamins, like especially vitamin B. So the microorganism that are used for these both things, and that's why it's termed microbioassay, and specifically for these two they are being used. The assay basically is carried out by comparison between inhibition of growth of microorganism produced by the reference material of known concentration, and measured dilution of test material. Especially this the thing is for so comparing the zone of inhibition that is produced by the both antibiotic, the test antibiotic as well as the standard antibiotic. Um, so that is being compared and then evaluating the that how much test antibiotic is active or potent or effective in inhibiting the growth of microorganism in the petri dishes or in the test tubes. So this definition of microbioassay, that the assay carried out by the comparison between the inhibition of growth of microorganism produced by reference material of known concentration and mere dilution of test material. That's for the antibiotics. Whereas if the same thing you want to know about the vitamins, it would be growth zone. So in case of antibiotic, zone of inhibition of growth, whereas in case of vitamins, growth promotion zones are checked, are compared, are measured. So this is the both are opposite in terms of the responses. The antibiotic used to inhibit, used to kill microorganism, where our vitamin are certain um, growth promoting agents in humans as well as in certain microorganisms. They need vitamins for their growth, so they can promote the growth. So the response is somewhat opposite if you are using antibiotic, if you are using vitamins. So in both responses, says the response is measured by diffusion in case of solid phase or medium, or turbidimetric method in case of liquid or broth medium. So whatever the growth inhibition or growth promotion zones are checked, basically zone is checked in the case of solid phase or when medium is used, and solid phase medium is used, whereas the turbidity, increase in the turbidity in case of vitamin, decrease in the turbidity in case of antibiotic checked when the liquid or broth medium is used. So the references for today's lecture are in front of you. That's all for today's lecture in theory class. So pharmaceutical quality management lab content. So object for today's lab is the determination of moisture content of sample, that is starch by oven drying method. Requirements for this test include sample, that is potato starch, china, or any evaporating dish available to you have, weighing balance, and oven for drying. So procedure says take 5 to 10 grams of sample in a china dish or any sample evaporating dish. Dry it at 120 degrees centigrade for 4 hours in an oven and reweigh. So calculate the moisture content of sample by formula. So the percentage moisture content is equal to W2 minus W3 upon W2 minus W1 multiplied by 100, where W1 is weight of container or your china dish with its cover. W2 is weight of container with its cover or lid and sample before drying. And W3 is weight of container with lid and sample after drying. So official limit for potato starch is that it loses not more than 14.0% of its weight. So whatever weight of sample is there, it will lose not more than 14% of it. So this is the official limit or range for the moisture content for the potato starch.